Yeah, so hi everyone, my name's Craig Stehn. I'm in the Ecosystems and Threatened Species team, the Department of Climate Change, Energy, the Environment and Weather. I'm going to talk today about some of the species conservation actions we're implementing for myrtle rust affected species in New South Wales. So I come from Gumbaynggirr country on the mid-north coast of New South Wales. The work I'm talking about today covers multiple Aboriginal nations up and down the coast, so I'd just like to acknowledge that and pay my respects to elders past, present and future. And also thank the New Zealand team and Jen in particular for organising this conference. It's been great. So before I start, I'll just give a bit of background on saving our species. It's our flagship threatened species program in New South Wales. It's been going for nearly 10 years now. We've had quite good funding over that period, 175 million. We're up for review at the end of next year and we're hoping we'll get another 5 years after that. So the SOS approach is very much species specific. So our projects are targeted at a species and there's a set outcomes and monitoring targets for those projects. We also have a real focus on engaging with experts and developing partnerships for projects. And I think the myrtle rust work is a real classic example of that. Across New South Wales, we've got about 80 people working on SOS, so we've got a fairly large team. So I think a lot of people probably know most of this already, but myrtle rust arrived in Australia in 2010. We have a lot of host species, so over 390. For context, that's about 17% of all of our native Myrtaceae in Australia, so it's a fair proportion. At least 43 of those are thought to be fairly severely affected, and they're listed as priority species under the National Action Plan. On the map there, you can see the distribution of those priority species. So you can see Queensland and New South Wales have the majority. And that red sort of zone, while it's not a definitive map of the distribution of myrtle rust, obviously, it's the area where myrtle rust is probably the most well established and most widespread. And you can see there's a really strong overlap there. So in New South Wales so far, we've had 4 species that have been listed as critically endangered because of myrtle rust. I'll be talking mainly about Rhodamia rubescens and Rhodamia civioides, but we've got Lengua, the species main range, which has been the focus of a previous talk, and Rhodamia maydeniana, which is Rhodamia that's limited to the north coast of New South Wales and southeast Queensland. So I'll just run through those first 2 species. Native guava first. It's a sort of a large shrub, up to about 12 metres tall probably. It's got a very widespread distribution from just north of Sydney up into southeast Queensland. Across that distribution, it's really common and abundant, prior to myrtle rust at least. Very common in littoral rainforest, so rainforest just behind the coastal system. It's declined to a point where it's now listed as critically endangered in New South Wales, Queensland, and at a Commonwealth level. And, you know, this species is was such a fast-growing, hardy pioneer species. It was often used in, you know, dune care and land care plantings. It was just such a hardy species. Unfortunately now, what we tend to find is most of the individuals are small root suckers. You can see in that photo there, I've got one, you'll see fields of these small suckers and they come off the root systems of what would have been a large adult plant that's often died and fallen over now. We know these sucker fields can persist for a long time. We've got locations where they're still putting up suckers after 14 years. Occasionally those suckers will get up and get to a metre and a half high. We'll see flowering and fruiting, but a lot of those fruits don't contain viable seed. So seedlings are just not observed in the wild. They're all these root suckers. Scrub turpentine, Australasian is similar. It's a much larger plant. It's a medium-sized tree up to about 25 metres. It's even got a more widespread distribution, occurring from pretty well, not far from the Victorian border up into the southeast Queensland. This plant occurs in heaps of different plant communities. A lot of rainforest and wet sclerophyll communities will have this species in it. So it also has declined to a point where it's been listed as critically endangered in New South Wales, Queensland, and at a Commonwealth level. This species too was a really hardy, fast-growing pioneer. Most nurseries doing native plants on the East Coast would have grown this species. It was planted out all the time. So what we're finding with rhodamia now is most of the plants we see are probably between 2 and 6 metres tall. One thing that's promising though is there's variable susceptibility in this plant. We find some plants in the field that appear to be quite healthy, others quite infected. It still persists across most of its distribution, but the numbers are just much less. There's one private land site that I know of that had 500 stems when I went there. It's still there, but no really of the quantity. We do get flowering and fruiting. Last year in particular, we had a really good year for flowering in this species, but most of the seed doesn't contain, most of the fruit doesn't contain vital seed. And we think that that's probably because of a lack of effective fertilisation. So one thing that's common with all of the critically endangered species, and also a lot of other species on that priority species list, is that they're suffering from rapid decline. So declines that have been so rapid that a widespread species can be listed as critically endangered. Flowering and seed production we know is impacted by myrtle rust, and seedling recruitment is thought to be basically non-existent. So the plants we're seeing out there now are likely the last natural recruits of these species in the wild, and they're gradually disappearing. So because of this, the conservation response is really focused on 4 key areas. Securing wild germplasm has been the initial focus, and we're trying to establish representative ex situ collections. While we're doing that, we're also doing genetic sampling, and that genetic sampling so that we can have some insight into the genetic variability that's in the wild population, but also our ex situ populations. Then we're looking at trying to identify resistant genotypes, and that's done through field observations as well as assays of seedlings in the collection. And then the step that we're hoping to get to is to use tree breeding techniques so that we can transfer that resistance into other wild genotypes. I'll just sort of go through that in a little bit more detail. So the ex situ and genetic studies have been delivered in partnership with the Royal Botanic Gardens in Sydney, or the Botanic Gardens of Sydney as they're called now. In particular, the plant bank, research and horticulture teams there. So the germplasm collections have focused on semi-hardwood cuttings for Rhodamia rubescens, and for Rhodomertis sidioides, we've also used a lot of root suckers because they're quite easy to dig out. And that core conservation collection's held down at Mount Annan, quite a large collection. We've got a photo there of Veronica in front of just a small portion of that collection. And the genetic studies tell us too that those ex situ collections that we have are genetically representative, and we know they're also geographically representative of the distribution. Those ex situ collections also form the basis of our resistance screening and resistance breeding work, and so far that's focused on Rhodamia rubescens. So over the past year, the team down there has been focusing on reproductive studies, looking at hand crosses between resistant and susceptible individuals, and then propagating those seedlings. And that work's been led mainly by Karen Summerall out of Plant Bank. So from those seedlings that have been grown from those crosses mainly, we've inoculated and performed assays now on just over 740 Rhodamia rudescens. And this work's taking place out of the Plant Breeding Institute at the University of Sydney. So basically that involves spraying these plants with a fine mist of Australasian CDI spores suspended in mineral oil. They spray the plants, they then maintain them at 22 degrees, and after 14 days they're assessed for disease. So what we've found with Rhodamia rudescens, which is really encouraging, is that 50% of those inoculated plants do come back as having some level of resistance, and some of them are coming back as highly resistant. So once we've done those assays on those seedlings, we've now got a collection of plants that we believe have some level of resistance. So we've planted out 122 of those plants so far to test them under field conditions. One of those collections has gone out to Minimi IPA, so Indigenous Protected Area. It's on the north coast of New South Wales. We've been working with the Minimi Rangers on many projects for quite a while now, and we're partnering with them for this work over the next 4 years. They're planting out, maintaining and monitoring these plants. And yeah, it's great to be able to continue that partnership with them. We've also got plants down at the Botanic Gardens at Mount Annan. 19 were planted in Coffs Harbour, and I've got 5 in my front yard as well. So what have we seen? The initial plantings happened in June 2022. They were really an opportunistic planting. We had 15 cutting grown plants that had gone through the assays and we thought were resistant. I brought them back up to the north coast. We planted 10 out at Coffs Harbour Botanic Gardens, and we planted 5 in Dunedin Road. Most of those did go on to develop heavy infections, but there's photos of 2 of those original ones up there. So that one on the left, it is covered in new flush every year. There's plants all around it with myrtle rust on it. We haven't seen an infection on that plant yet. The one on the right was planted at the same time, and that photo was taken probably 6 months later, and you can see it's totally defoliated. So there's obviously a high load of spores in that area, but that plant is definitely coming through a lot better. So the second planting took place earlier this year. We planted 107 seedling grown plants, and they've gone out to 3 different locations. So far, only 3 of those have come down as being susceptible to myrtle rust. It's still early, we haven't done much monitoring, and we're coming into winter, so that may change, but I think that the assays probably work better in the seedling grown plants, so we're hoping to see a lot more resistant plants in that group. Ooh, don't know what's happened to that photo. Anyway, over the next 4 years, we'll be monitoring these plants every month, and we're just trying to, I suppose, confirm those assay results, look at how that observed resistance changes as the plants mature, whether or not there's any change when they flower and fruit, and collecting information on flowering and fruit set, and hopefully also collecting open pollinated fruit. And we're using an app called EpiCollect to do this, which is a good free data collection app. So going forward, I think one of the biggest challenges we've got is trying to scale up the work we're doing, so the susceptibility screening and the resistance breeding. So at the start of the talk, I mentioned that there were 43 plants that were considered priority species. 22 of those are actually already listed as threatened. They're either listed because of declines from myrtle rust or because they were threatened prior to the arrival of myrtle rust. So we've got 22 species there that are potentially in, a pretty bad situation. As I mentioned, most of the work we're doing so far just focuses on Rhodamia rudescens. So obviously to expand that work to target these other at-risk species, we really need substantial funding for staffing facilities, and it's something we don't have at the moment. The other thing is we also have all these other species that aren't listed yet that we're quite concerned about. Gossia hilii is actually in the process of being nominated. It's being assessed in New South Wales and at the Commonwealth level now. But then we've got other species like Archaeorodonata speculari, Badecospermum, where in some parts of their range they seem to be coping okay, and then in other areas they've become locally extinct. We've also got situations where there are community level impacts because we have potentially all these three species, but also all of my target species that I'm looking at, the Rhodamna rhodescens and the Rhodovertis sideroides, all co-occurring on one site, and they're all, basically disappeared from those plant communities. So you'll see the entire mid layer of a area that's just dead. And I think the other thing that's really worth keeping an eye on is biosecurity. So we know there are other strains out there, they could arrive in Australia. We also have areas in Australia that are free of myrtle rust, so Lord Howe Island's one location, but southwest Western Australia as well. And I think Lord Howe Island's a good example of how that vigilance has paid off. Myrtle rust was detected there in 2016, and the local community jumped onto it straight away, and it was eradicated. 2023, it did turn up again. Same thing, it was picked up really early by a community member who had a nursery background or something unusual on one of his Christians, reported it. Within a couple of days, they'd mapped every single metaceae in the settlement area of Lord Howe, and they were able to eradicate it again. So I think that vigilance pays off. So that sort of wraps up the talk I want to give today. I really want to acknowledge all of those project partners. They put in so much effort and time into this project. And if anyone's interested on myrtle rust in Australia and wants to find some good resources, I encourage you to go to the Australian Network of Plant Conservation website, and that QR code will take you there. Thanks.